What's up, everybody? My name is Dimitri Kofinas, and you're listening to Hidden Forces, a podcast that inspires investors, entrepreneurs, and everyday citizens to challenge consensus narratives and to learn how to think critically about the systems of power shaping our world. My guest in this week's episode is Dr. Scott Gottlieb. Dr. Gottlieb is a physician who served as the head of the FDA from 2017 to 2019. He currently serves on the boards of Pfizer and Illumina and is a regular contributor to CNBC, where he has really become, I think, one of the most recognizable faces in America. His recently published book, titled Uncontrolled Spread, examines how the coronavirus and its variants overwhelmed our national defenses and the steps he thinks we need to take in order to better protect ourselves and our country from the next pandemic. There was so much that I wanted to discuss with Scott today that I literally could have talked to him for 5 hours and still not gotten through all of my questions. In fact, I've recorded a lengthy afterthought segment in place of the regular overtime this week in order to share my own thoughts about the conversation, the research that went into it, what I learned from doing it, and what I think are some of the most interesting avenues to explore in future episodes with other guests on this topic. That said, we covered a lot with Scott today. We spent the first half discussing some of the systemic failures that Dr. Gottlieb thinks exacerbated the scope of the pandemic, as well as the economic costs associated with the various lockdowns and shutdowns that were imposed in the very early days of the outbreak. We also got into quite a bit of detail about not only the biological mechanics of the virus, how it spreads, and what we've learned about best practices for protecting ourselves and those around us, but also about the various vaccines, their effectiveness, how they work, specifically how the mRNA vaccines work and what makes them so promising, as well as the risk of taking them, both known and unknown, and how to evaluate those risks when making your own decision about whether or not to get vaccinated. We also discuss alternative treatment options like ivermectin and whether or not Scott thinks there is any censorship going on in the mainstream media around talking about these types of alternative treatments. As I mentioned at the top, this week's premium content is an afterthought segment that I recorded right after this conversation, where I reflect on not only what we discuss in this hour, but also many of the things that we didn't get a chance to talk about, like the lab leak hypothesis, the issue of mandated vaccinations and the argument for and against them, the future of sequencing and genomic epidemiology, new antiviral drugs, the risk of new BSL-4 facilities cropping up all over the world and the existential risks that they pose, and so much more. If you enjoy the free content that we produce every week, I encourage you to take the leap and become a premium subscriber if you haven't already. There's no commitment, you can cancel at any time, and the entire library of subscriber content going all the way back to episode 1 becomes instantly available to you, including the overtimes, afterthoughts, transcripts, and rundowns, depending on your tier. The rundown to this week's episode is a nearly 30-page compendium full of notes, questions, images, and other materials that I put together to help me and you through this conversation. So without any further ado, please enjoy this week's educational episode on the COVID-19 pandemic and its future with my guest, Dr. Scott Gottlieb. Dr. Scott Gottlieb, welcome to Hidden Forces.
Thanks for having me.
No, it's awesome having you. Before we begin our conversation, I just wanted to tell you how much I appreciate the work that you've done the last 2 years, not just because you've been a really phenomenal educator, and you have, but I think that you are an example of the kind of even-tempered, scientific, genuinely scientific-minded people that we need to have, not just in policymaking, but even in journalism. So I really do appreciate that.
Thanks a lot. Thanks for having me here today.
Sure. So a big part of your book, which I've mentioned to audiences, the book is called Uncontrolled Spread. I would say a big part of the book is a sort of contemporary history of the COVID-19 pandemic that gives readers a broader context for why things progressed the way they did, why we got the outcome that we currently have, and what we need to do in order to improve our preparedness for what you consider to be a national security threat on par with asteroid impacts or radiological attacks. Is that a fair summation of the book, and what would you add to that overview?
Yeah, look, I think that that's a fair summation of the book. I talk about the need to look at public health preparedness through a national security lens, and the reference was to radiological attacks or meteor showers that could be an extinction level event. Is that we make preparations against low probability but high impact events in other facets of our life. Nobody expects a dirty bomb to go off in a city or a large meteor to strike the earth, yet we make preparations for those events because they could be catastrophic. We didn't make adequate preparations for a pandemic, yet a pandemic was a more likely event than those low probability events. It's still a low probability event that you have a global pandemic that's this devastating, but we didn't make adequate preparations for it. And we need to look at it as part of our national security preparations. And if you're looking at pandemic preparedness through that lens, you do a lot of things differently. You get our national security agencies more engaged in the overseas mission of trying to identify these kinds of threats, and you get domestic agencies much more engaged in making preparations that create a hot base of preparedness. So you have things ready to go, and we didn't do that.
So we're going to have a chance to talk about some of those different solutions or ways to prepare towards the end of the conversation. But was that your primary motivation in writing the book, to get people aware about what we can do to prepare ourselves for the next pandemic?
It was. I spend time telling stories and sort of recounting what went wrong, but with an eye towards the sort of conclusions, the ideas on what we need to do differently. So the way I set it up in the book was focusing on aspects of the story. I didn't tell the whole COVID story. I told aspects of the story that identified the weaknesses that I think we need to solve. I thought the book would be coming out. Look, I'm very grateful for the reception the book has had, but I thought the book would be coming out in the middle of the legislative debate about how we create the plan for the future. And I'm surprised and a little bit disappointed, just not in the sort of timing of the book publication, but disappointed in the policy making that we really haven't engaged that discussion yet. You don't see a commission formed. You don't see Congress taking up legislation. You don't see, the administration putting forward detailed plans. They put forward a strategic plan, sort of a top-line plan, but it's an outline of a plan. We haven't taken that up yet in earnest. And I suspect it's because it might be too early. We're still in the middle of the current pandemic, and people, have a perspective that we have time to work on this. That's probably right, but we don't have endless time. We face constant threats from new pathogens, and the fact that we haven't put flu immunity in the population probably for 2 years sets us up for a pretty bad seasonal flu season, and maybe even makes us more vulnerable for a pandemic strain to gain root. So in many ways, the risks are mounting, and I think we need to start having the discussion. If we wait too long, people lose sight of what happened, and then maybe they're less willing to engage dramatic action to do things differently going forward.
Right. To that last point, I think that is also a characteristic of how we've approached many of the recent crises. We fight them sort of with overwhelming force while they happen, and then we really kind of wash our hands of them after they're over and don't really do the proper kind of retrospective that we need to do in order to be better prepared for the next one. Your point about flu, are you suggesting that because of the mitigation measures that we took, like social distancing, for example, and some of the lockdowns during the course of COVID, that because we took those measures, we didn't really see a flu season like we would have seen in previous years. And as a result, people haven't built up the kind of immunity that they would have gotten from that pathogen as well as other ones potentially?
Yeah, that's right. We didn't have the mitigation that we did proved very effective against flu. It proved effective against COVID. It helped reduce the spread of COVID, masks, hand washing, distancing. But COVID spreads, we now accept, even though this was obvious to a lot of people earlier, COVID spreads primarily through aerosols. So COVID spreads from one person to 30 people. It's a situation where you get the wrong person in the wrong place at the wrong time. You have someone who's pre-symptomatic, maximally contagious, in a confined environment where they're talking loudly or singing with poor air filtration. That's the circumstance which COVID spread. Flu spreads from one person to two people. It spreads primarily through droplet transmission. So the mitigation, which was actually designed to help stop the spread of influenza, that's where a lot of these practices come from the 2005 pandemic plan, which was geared entirely towards flu. The mitigation works very well against the flu. And so what happened last year? We had basically no flu season. This year, there's some indication it's going to be another mild flu season. A little too early to conclude that, but there's some indication of that. So if we have 2 years of a very mild to no flu season, you have 2 years of no flu immunity put into the population. And then if in year 3, you get a mismatch between the vaccine and the actual circulating flu, which is possible because you're not going to have a lot of flu around the world to actually gauge very precisely what the prevalent strain is going to be. I mean, the way we sort of decide our flu vaccine is we look at what's circulating. If there's not a lot of flu circulating, you don't really know what could become the prevalent strain in the wintertime. So if we have a mismatch between the vaccine and the circulating strain that gets hold next year, you could have a really bad flu season. I mean, we're going to be in for a whopper of a flu season at some point on the back end of all this. And, that's also, you have a really bad seasonal flu season, it could have the contours of a pandemic strain of flu in terms of how it spreads and the level of death and disease it causes. So, the idea that we have endless time to wait before the next pathogen strikes us in a similar way may not be the case.
That's interesting. I might want to ask you more about that when we talk about mRNA vaccines, because I'd be curious how that technology could be deployed for the seasonal flu, because I don't think actually we've had an mRNA vaccine produced for flu, for influenza, for the population up until now, but you might correct me if I'm wrong. In the book, you write that we, quote, prepared for the wrong virus. And your point being that we had prepared our mitigation response protocols were built around flu. So my question is, how does SARS-CoV-2, COVID-19 in other words, how does that differ from strains of influenza? And how do those differences affect or inform how the public health agencies should go about establishing mitigation protocols?
Yeah, I think that the right way to prepare for a future pandemic isn't to prepare against specific pathogens. We've always tried to focus on specific pathogens, and we prepared for flu, and we prepared for special pathogens like anthrax and smallpox, and pathogens that would be used in a deliberate attack. And that was sort of the contours of our preparedness, our plans against biological risks. I think what we need to prepare for, we need to look at all the pathogens that could potentially threaten us and derive the characteristics of those pathogens and prepare countermeasures for those characteristics. So a pathogen that spreads through droplets, one that spreads through aerosols, pathogens with long incubation periods where diagnostic testing is very critical, pathogens with short incubation periods where testing and tracing isn't going to be as effective because there's a short incubation period. So by the time you identify a case, there's multiple generations of spread. Pathogens that spread through asymptomatic transmission and those that don't. We need to sort of catalog the range of characteristics and have a plan for how you employ countermeasures against those characteristics. Because even if the next pandemic is flu, it might be a flu that has characteristics that are very different than seasonal flu. Coronavirus has characteristics that are very different than seasonal flu. And I'll give you a practical example of this. I was part of the pandemic planning for influenza. I was there in 2005 as deputy commissioner of the FDA when some of these plans were written. I wasn't a central player, but I certainly was involved and was cognizant of what was going on. That plan was entirely focused on influenza, and it was focused on a very specific strain of influenza, H5N1. We were worried about a bird flu. And we did tabletop exercises. We drilled for a pandemic contingency, and we did those in the Trump administration too, and the Obama administration did it. This was constant action. Every administration was planning, but they were planning for an influenza pandemic. Well, in those plans and in those tabletop exercises, having diagnostic tests that could be widely deployed was never really a critical feature. We never struggled with it for a couple of reasons. Number one, in an influenza pandemic, flu has a very short incubation period, 3 days. So someone's exposed and they get it 3 days later. So using testing to try to find chains of transmission isn't as effective because the chains of transmission are so short. Once you find a patient, they've already transmitted 2 generations. Also, patients aren't contagious until they're symptomatic. So you can identify them based on symptoms. Getting someone diagnosed with the infection when they're pre-symptomatic or asymptomatic isn't as critical. And the nation's installed base of flu testing would be adequate because in a pandemic, let's say you have a pandemic with a strain of influenza B, or let's say it's influenza A, every doctor's office in the country has a test that could differentiate influenza A and influenza B. You don't need to sequence it and figure out it's the pandemic strain. If a pandemic influenza B is spreading and a patient comes into your office really sick and they have influenza B, you assume they have the pandemic strain because there's going to be one strain of flu predominating. So testing was never a critical feature. And when we did the tabletop exercises, we never said, how do we scale massive amounts of testing and deploy testing sites around the country? That was never a question because we prepared for flu. We didn't prepare for a coronavirus, and we never thought we'd have a virus we would need to actually sequence using PCR to detect genetic sequences to actually identify it. We never thought we'd need to set up respiratory collection sites So all that infrastructure, all that planning had never been done, again, because we prepared for a flu.
So that actually touches on a few things all at once, and I'm going to try to pull out and put them in order. That touched on sequencing and genomic epidemiology and testing as well. So let's focus a little bit on testing, because what you hinted at there was that there are conditions in which you want to have massive testing so that you know what kind of strains you're dealing with in case you're dealing with mutations, in which case you want to get ahead of those with vaccine development. I presume that's what you were implying with that. But there's also something else, which is simply the fact that the way in which this virus spread was primarily through asymptomatic spread. And to go back to your point about the flu, the reason that public health officials never really considered asymptomatic spread as a major culprit in accounting for the high R0 score was, in fact, because they were working off of flu mitigation models. Talk to me a little bit about the importance of testing and the consequences of lack of testing and our inability to scale testing up, not just the actual development of tests, not just their rollout, but also the protocols around their use, which tests should be used for what environment, et cetera. Where would we be today if we had approached that differently?
Yeah, so in the book, I spend a lot of detail on what went wrong with the testing. Probably the most detailed chapters in the book are the blow-by-blow of what happened at the CDC, what was going on at FDA. And I felt it was really important to get that history told. I spent a lot of time researching that, talking to sources inside the government to get that history down on paper. And the reason is because, number one, the lack of testing was at the root of a lot of our early and our later failures and left us in a much more vulnerable position. But I also think what happened with the test was sort of a metaphor for the broader lack of preparedness. The lack of being able to scale a diagnostic test was the most obvious example of a more systematic lack of preparedness across the country. So what happened with the testing? Early on, as everyone remembers, we didn't have a diagnostic test. We really never had enough diagnostic testing until we got into the winter time. And that was a failure of leadership way back in the beginning, where when this all began, there was this presumption that CDC was going to develop a diagnostic test and mass produce it and distribute it to labs. CDC had never done that before. CDC had never developed a diagnostic test and distributed it beyond the public health labs. There's 100 public health labs in this country, each able to do 100 tests a day. It's 10,000 tests. That is a fraction of a fraction of a fraction of the amount of testing we needed. So the political leaders who thought that CDC was going to develop this test and deploy it, even if they had done it perfectly, which they didn't, we never would have had even nearly enough testing. And of course, CDC famously contaminated their lab. I talk about how in the book. They were testing samples from patients in the same facility where they were assembling their test kits. It looks like virus got from the patient samples into the test kits. They contaminated them. The test didn't work. What needed to happen was back in January, someone needed to say, this looks really bad. This looks like it could become a pandemic. We need to get the major manufacturers and large clinical labs in the game. We need to, hit the red button and do an all-of-the-above approach to get as much testing into the market as possible and backstop the financial risks that these companies would be taking so that they're willing to make the investments. Once they get going, once the big commercial manufacturers start developing test kits at scale, that's a 6-week process. I mean, maybe you can crash it and do it in 4 weeks. That's ambitious. It's probably 6 weeks. It might be a little bit longer. So we needed to start in January if we wanted to have massive amounts of testing available in March. As it were, that phone call never got made really until the end of February. And so we never had enough testing. And so what did that mean, that we didn't have enough testing? Well, number one, we got heavily seeded with infection all across the country because we couldn't detect it. So people were presenting with flu-like symptoms, testing negative for flu, and we didn't know what they had. And we couldn't even diagnose them to properly isolate them on an assumption that they had coronavirus. So you had people presenting with coronaviruses and no one knew it, and you had the country heavily seeded. The other place where the testing created a major fail was that not only didn't we know where the virus was, but we didn't know where it wasn't. So when New York was on the brink of collapse and the healthcare system was breached, and we had to do the stay-at-home orders in New York and Chicago and New Orleans and Boston and Detroit and San Francisco, those cities clearly were on the brink of collapse. I mean, New York's healthcare system became COVID only. Their health capacity was breached. They had to set up tents and a hospital center in Javits Convention Center. But Bozeman, Montana didn't have a lot of spread, and Houston, Texas probably didn't. And all across the South, the virus really hadn't reached a lot of those cities. But instead of targeting the mitigation to the places that were engulfed with virus. We did the 15 days to slow the spread, followed by the 30 days to slow the spread. We did a simultaneous national shutdown for a virus that has ended up being very regionalized in its spread across the country. This is a big nation. It's a diverse nation. The virus hasn't inflamed the whole nation simultaneously. I mean, look at the Delta wave. The South was engulfed with Delta. New York didn't have a lot of Delta infection. Now New York is starting to have more Delta infection. So this has been a highly regionalized spread through most of the pandemic, with the exception of last winter when we had more of a confluent epidemic across the country. But because we shut down the whole country, when the virus eventually got to the South, everyone down there said, we're not shutting down. We shut down in the spring. Mm-hmm. You told us to. We listened. We probably didn't have to. And we're done with that. And so the political leaders lost the capital and the clout they needed to implement the hard mitigation when the virus finally arrived. Because we didn't have a test to say, you know what, in Texas right now, there's not a lot of spread. So let's hold off on the shutdown order and let's use testing, tracing, and quarantine as a way to contain the spread, because there were parts of the country where you could still use what we call case-based techniques. You could still use testing and tracing and quarantine of sick people as a way to control spread. You don't have to do the population-wide mitigation. The population-wide mitigation comes in only after you've lost control. And there were only certain parts of the country that we had lost control of, but we didn't know that. We were blind to the spread, all because we didn't have a diagnostic test that is literally, tens of dollars to produce.
Mm-hmm. So how much of the outcome that we got was contingent upon the White House and lack of leadership, and how much of it was institutional? Because in the book, you talk about this in terms of a system-wide failure across government institutions. So how do we think about what went wrong and how much of it was just bound to happen based on our lack of preparedness?
Well, look, the institutional aspect of this is that CDC, which had the ball, CDC was in charge, is very insular, very territorial. So if you were a major manufacturer and you wanted to make a diagnostic test, you needed access to the sample of the virus. Well, CDC wouldn't give it to you. CDC actually didn't provide access to samples of the virus till the end of February. So if you said, okay, CDC won't give me a sample of the virus, so I can't design my own test, maybe I'll just copy the CDC's test design. Well, you couldn't do that either, because CDC said that nobody can copy CDC's test design unless they sign an intellectual property agreement with the CDC, transferring all the rights and patents to CDC. And the big companies looked at these big legal documents that CDC was putting in front of them, and they're like, this is going to take weeks to negotiate. And I talked to companies over this time period. They said, look, they're making it complicated. We're going to wait and see. They say they have the ball. They're making it complicated for us to scale up manufacturing using a test design. They won't let us design our own test. So we're just going to wait and see. So CDC, they said, we have this, and they weren't sharing. Okay. And that's cultural and institutional. Political leadership, someone needed to pick up the phone, as I said, and say, we're going to get these companies in the game. I'm going to break through all this red tape, and I'm going to get 3 major manufacturers to start manufacturing tests at scale. And there were plenty of times that those phone calls got made when I was in government, when we had a public health crisis and needed something. Certainly not on this scale, but those phone calls get made. It didn't get made in this instance. Was that the White House's fault? I think it's hard, and I don't let the White House off the hook in this book. You read the book, I'm critical of things that happen, but it's hard for a political appointee in the White House to have enough awareness of how things work to know how to make that call. That really has to happen in the Department of Health and Human Services. That's really in sort of the Secretary's immediate office, because that's where you have the understanding of how the agencies work and how they work together and what the capabilities are. There were people telling the White House that they had to get the diagnostic manufacturers in the game. The White House at one point took a meeting with the major manufacturers to try to coax them into the market. But that's different than getting, the operating divisions and the regulators and the secretary on the phone with those manufacturers who could make assurances and, work more closely with them. This has to happen at a level below the sort of White House political leadership, and it just didn't happen.
So does that mean that we need something like a JSOC, like a Joint Special Operations Command institution, to do this at the institutional level, so you don't have to rely on the executive branch to sort of make those phone calls on a regular basis and create interagency interoperability?
You need something that can bring better coordination across the different operating divisions. I talk about the idea of a sort of a JSOC approach, having something that can coordinate in a crisis. There's definitely coordination that has to happen across the different components of government, But what we're talking about here with the diagnostic test is coordination that had to happen within HHS. And we were supposed
So within
to,
the actual institution itself?
within the Department of Health and Human Services. And we were supposed to have solved for that when we created the Assistant Secretary for Preparedness and Response. I mean, we created the ASPR, this new office that was this emergency response office inside HHS that was supposed to be the office that brought the coordination across the different operating components of HHS in the setting of a crisis. So it didn't work. I mean, we had kind of thought of this and it didn't work. There was coordination that has to happen across the government. So you have to get like FEMA working with HHS, working with DOD, but there's also coordination that has to happen in HHS. And what we're talking about here with the lack of a diagnostic test is coordination that didn't happen within HHS. So there was a failure on a failure in the setting of this crisis, at least as it related to the early generation of diagnostic tests. I think a lot of it was sort of this misplaced confidence that CDC had the ball, and CDC not being able to sort of self-identify that they weren't going to be able to keep up with this. And I also think that there was, when you talk to people who were involved in this, I quote some of them in the book, they didn't expect this coronavirus to move this quickly. They didn't expect it to be this raging pandemic that, engulfed the world with this speed. The plan for rolling out the diagnostic test, and I got a copy of it, and I talk about it, was a 6-month plan. They had this 6-month timeline of how they would go through this iterative process to scale the production of a diagnostic test. We didn't have 6 weeks, let alone probably 6
Well, what
days.
accounts for that? Was there not enough information at that moment in time to understand that there was a sufficient risk, that this was going to have a high R0 score and that it was going to spread rapidly?
What I was told was that there were people who thought that this was going to be more SARS and MERS-like, which didn't spread as aggressively between people. So yes, exactly, the R0 would be lower. But this was at the point of...
But we were seeing images out of China.
Exactly.
Right. So
exactly. Right. So this was happening. This conversation was happening by the end of January when China had just locked down the Hubei province. I mean, they locked out, they crushed their economy to get control of this virus. That should have been a pretty clear indication that they were seeing a velocity of spread that scared them. There was enough information at that point to have a different judgment, but there was still a lot of confusion. Look, it's a fog of viral war. The information wasn't perfect. I think that there was enough information coming out of China that at the very least, even if you think that this could be something that doesn't spread with that kind of velocity, there was enough information to say maybe we're wrong. We should prepare for the worst at that point. Even if you didn't believe this was going to have an R0 of 3, there was enough information suggesting that this could be highly contagious based on what was going on in China that you said, okay, but let's just prepare just in case we're wrong.
What do you think that's about? Because we see this kind of institutional dysfunction across other areas of government. It's not unique to the health agency area.
It's a hard question to answer because, why did people believe the rosy scenario? I think that that's sort of the temptation. It's very hard when you're in these jobs to lean out too far and take aggressive action against a theoretical risk and be wrong. I'll give you an example that will sound small, and I don't mean to equate this to what these officials were dealing with, but right before Thanksgiving in 2018, I recalled all the romaine lettuce from the marketplace because there was an outbreak of E. coli in romaine lettuce, and it was literally 2 days before Thanksgiving. And so this was a national story. I got tons of criticism. Why did we do that? We did that because we knew that people were getting sick actively from eating romaine lettuce. And there was only one portion of the country that was harvesting romaine lettuce at the time. It was the Salinas Valley in California. So romaine lettuce is grown in different parts of the country, but different parts of the country harvest at different times of the year. There was only one part of the country harvesting at that point. So we knew that if we recalled all the romaine lettuce from that region, that was the only place where romaine lettuce was coming from. Clearly, the outbreak, we surmised that was the source of the outbreak. It was an active infection. So by recalling the food product, we could probably thwart subsequent infections.
Because it was effectively a sample size of what you knew was going to be
exactly.
Right.
Yes, we were able to isolate it very quickly. It's very rare with a foodborne illness that you can isolate the source that quickly and intervene while the outbreak is still going on. Most of the time when we find out about outbreaks of foodborne illness, it's well after the fact. The food's been in the market, everyone's eaten it, everyone's gotten sick, and it's almost like an after-action report. It's very rare that you can catch it in the middle of it. So by doing that, we thought we were able to stop people from getting sick. We were right. It was from the Salinas Valley. There would have been more infections if we hadn't taken action. But if I had been wrong and I had recalled all the romaine lettuce and hurt the industry in the Salinas Valley, that would have colored the rest of my tenure as FDA commissioner. I would have been before hearings in Congress.
Right.
It would have been hard for me to make any more food policy. So it's very hard to lean out too far, take aggressive steps that are going to compel people to do things, cost money, maybe hurt industries, and be wrong.
Is that another way of saying that you think that policymakers are more risk averse today than they were in the past or in the recent past?
I don't know. It's hard for me to sort of gauge the temporal history of this. I think it's probably harder today than it was in the past because there's more scrutiny. There's more ways that criticism can self-generate, particularly from like social media, because you have people, you can hear the voices of the people who are affected by bad policymaking. But I think that that's always inherent in policymaking, that it's hard to take aggressive steps to try to head off or try to prepare for a theoretical risk that doesn't materialize. You're going to be criticized. I'll just give you another example. Back in 2005, when we were doing all the pandemic preparedness, we were diverting money away from more routine public health problems to help fund some of this pandemic preparedness work. And I remember being in meetings with scientists where we were berated. I mean, I was harshly criticized because they didn't think that a pandemic was imminent or posed as significant of a risk as we were worried about, and yet we were taking money away from other priorities. And those were some tough conversations. So it's very hard to prepare for a theoretical risk or a hypothetical and be wrong. In the case of the foodborne outbreak, we withdrew all the romaine lettuce based on a hunch, now a very informed hunch, that there was active infection going on and that people could potentially be harmed, and we could head that off. We were right, but we could have been wrong. And the recriminations for being wrong would have been significant politically.
Right. Well, that speaks to this trade-off between efficiency and resiliency. I mean, if we have perfect information, we can design the most efficient system. But resiliency speaks to the uncertainty, and we prepare for that by building redundancy in our systems and doing things that aren't necessarily going to be used. For example, stockpiling masks that may never end up getting used, or stockpiling or actually building production capacity and having it, as you say, hot and on the ready when it may in fact never be used. And that's what effectively insurance is. I want to pivot to the vaccines here just because I want to make the best use of our time as possible. One, I want to talk briefly about mRNA vaccines. If we had more time, I'd love to actually get into the science of it because it's fascinating. I actually wonder, in fact, this is the question I'm going to ask you first, which is if the public had been familiarized or been familiar with mRNA technology and its compressed development timelines from the outset, do you think it would have reacted with less skepticism to the early deployment of these vaccines? In other words, because everyone was used to thinking about vaccines in the sort of traditional, perhaps even analog sense of what they were, as opposed to where the technology is today.
Yeah, probably the novelty of the platform that was used to develop the vaccines contributed to some of the skepticism, the early skepticism and the persistent skepticism. But I don't know that for most people, it's sort of a highly informed view of this platform versus a traditional platform. Because when you sort of decouple what this platform is doing relative to like how we make flu vaccine, it's actually not that novel. I mean, it's the same basic principles of immunology. You're just doing things differently. I think it's more just a perception of something novel, and then that creates the ability for people who want to sow skepticism to point out attributes that sound unfamiliar. So, the idea, well, you're getting a gene sequence. The mRNA vaccines are a gene sequence, and then you see all this stuff on Twitter, they can integrate into your DNA. I mean, that's not how they work, but the novelty of the platform creates the opening to then sow that doubt. So yes, it probably contributed to it. The fact that these were fully synthetic vaccines was the first time we developed fully synthetic vaccines at scale.
Well, aren't they much faster to develop as a result of that fact?
Right, coming up with the vaccine construct is. So just to decouple this, with a traditional vaccine, and this is how the Chinese made their coronavirus vaccines, but let's just take the flu vaccine. What you need to do is you need to get the virus, you need to find a cell culture that the virus is going to grow efficiently in, and in the case of flu, it's usually chicken eggs. You need to tweak the virus so it grows in that cell culture. You need to grow it in large quantities. You need to inactivate the virus, and then you cleave off its surface proteins, and those proteins become the stock for the vaccine. In 2009, with the H1N1 outbreak, one of the reasons why it took us longer to get a vaccine than we thought it would was because we found out the H1N1 flu didn't grow efficiently in the eggs, so the yield was much lower, and we had to tweak the virus to figure out how to grow it in sufficient quantities. That's how a traditional vaccine's made. That's how the Chinese approach the production of the coronavirus vaccine. In the case of mRNA, what you start with is the sequence. You don't have to grow the virus. You don't even need the virus. You just need its sequence. And instead of growing the virus, what you do is you take the sequence, you put it in a liposome, and you use that basically as the vaccine construct. So what you're injecting into someone's arm isn't the protein that you want people to develop immunity against. You're injecting the sequence that codes for production of the protein, and then the body becomes the manufacturing plant. So instead of growing the virus in a cell culture, what you do is you give someone the mRNA sequence, and their cells take that up, and they basically produce the protein. Not the whole virus. It's not
the antigen.
Just the antigen, just the very discrete portion of the virus, the protein on the surface of the virus that you want to target your antibodies against. So in one case, with the old approach, you're giving the protein, and in the mRNA approach, you're giving the sequence that codes for the production of that protein.
It's almost like you give a hound the scent of a dead duck in this traditional way in which we develop vaccines versus an alternative case where you have some kind of like brain-computer interface and you give the dog the ability to simulate the scent, and then it's able to pick it up.
You're co-opting the body as the production factory for the protein that you want produced so that the body can then develop antibodies against that. But the reason why it's a much more efficient development process is because getting the actual vaccine construct, you don't need to isolate the virus and grow it and inactivate it and cleave it. You just start with the sequence. So you can literally do it in a matter of weeks. So the vaccine, the original construct for the vaccine, we developed very quickly. We literally developed it in a matter of weeks. The clinical development, the trials that were done, those were traditional trials. I mean, these were the largest outcomes trials ever done in modern times. So there was nothing short about the actual development program and the testing in humans. The only part that went fast was coming up with the actual initial vaccine construct. And that's because we used fully synthetic tools for the first time at scale to do that. I mean, this technology has existed for decades. We've used it for other things. But this was the first time we used it for a vaccine at scale.
How consequential was the choice of the spike protein as the antigen on which these mRNA vaccines would focus for the robustness and resiliency and staying power of these vaccines in the face of possible adaptability of this virus?
Yeah, very consequential. And that's where the earlier work that we had done on SARS-1 and even MERS came into focus. I mean, we knew because of prior experimentation with coronaviruses that the spike protein was the optimal target it was the immune dominant target. It was going to be the epitope that was most attractive for developing immunity against. If we hadn't done that earlier work, if NIAID and the NIH hadn't done that earlier work, this would have taken much longer because we would have had to do the experimentation to figure out that was the optimal protein to use in a vaccine. In fact, in the case of Pfizer, I'm on the board of Pfizer, as you know, we tested two different constructs. One was the full-length spike protein as the epitope, and the other was just the receptor binding domain, just the tip of the full-length spike protein. And we tested both and took forward the vaccine that was based on the full-length spike protein, which in retrospect looked like it turned out to be a fortuitous decision. As the virus has mutated, we're probably better off with a vaccine that includes more potential vaccine epitopes. So, there was still work to be done, but a lot of it in figuring out what the immunodominant part of this virus was had been done previously.
Right. And just to quickly summarize, the reason that I brought it up is because the spike protein is the pathway, the mechanism through which the virus enters your immune system.
Right.
And so creating immunity against that protein forces the virus to try and find some way around that, but that complicates things because that's the primary mechanism by which it infects you. So this might actually be a good opportunity to pivot into some of the more controversial topics that I wanted to get into with you, Dr. Gottlieb, one of which is vaccine risks. So there are a lot of opinions and theories that circulate about the vaccines. And in fairness, there is always going to be uncertainty. No one can tell you definitively that if you take any one of these vaccines that you aren't going to get sick or have an adverse reaction. And there are short-term adverse effects, many of which I think we know more about today because these vaccines have been around now for like a year or maybe close to a year and a half in some cases. And then there are long-term risks as well, about which we know much less. So with that preface, what do we know about the risks associated with taking these vaccines?
We have more data around this medical product than many, if not most, medical products, in part because, we now have a year and a half, almost 2 years of data from when the first people were put into clinical trials. But we've distributed 300 million doses in the US. We've distributed 6 billion doses of vaccine globally. So there's an enormous, and the information is getting collected very actively, right? That surveillance is very active on people who've been vaccinated, probably more so than with any medical product going on right now. So that's creating a very robust data set. The profile looks, clean. I mean, if there were things happening at high rates, even if the vaccine was increasing the risk of something that ordinarily occurs in a population, you would expect to start seeing signals now. There have been signals, for example, the risk of myocarditis and pericarditis in young men who were vaccinated, where you saw an increased incidence. Unclear if it's vaccine related, but certainly something that people should be aware of, that people should factor into decisions about how to safely use the vaccine, that doctors should be aware of in terms of appropriately monitoring patients. But there haven't been a lot of strong signals that have become obvious. And the ones, even the rare events, like the risk of TTP with thrombotic cytopenia with some of the vaccines, those are extremely rare events, and we were still able to detect them because the surveillance has been so strong.
What about long-term risks that won't emerge for many years to come? How do people evaluate those unknown unknowns?
Well, most risks from vaccination manifest within the first 3 months.
That's typically the case with vaccines?
Typically the case with vaccines. What you're postulating is the theoretical possibility that there is a latent risk that's not manifest for many years because of exposure to the vaccine. And it's hard to sort of postulate what that would be scientifically, what the mechanism would be of the vaccine inducing some latent risk, where what it probably would be, just sort of talking in theoretical terms, the hypothesis would be that exposure to the spike protein creates some immunoinflammatory response that will increase your propensity to develop something X number of years out. Right? That's kind of what you're postulating. And I'm not trying to put words in your mouth.
No, that's exactly what I'm postulating. Let's say exactly you correct.
Let's say you as an individual have a predisposition for some neurological disease, or not you, but someone. And exposure to the spike protein creates an inflammatory response that sets in motion a biological event that then makes that low probability event that might have happened to you higher probability. If that's the case, if somehow it does that, then presumably it's not going to be exposure to the vaccine that does it, the vaccine construct. It's going to be exposure to the spike protein.
Right.
And if that's true, then the same risk would be manifest if you got infected with COVID, and maybe more so. And in fact, like for the flu vaccine, we see a very low incidence of Guillain-Barré, a neurological event in the setting of flu vaccination. We see a higher incidence of Guillain-Barré in the setting of flu infection. And why? Because the epitope being used in the vaccine is what's predisposing or what's triggering that immune reaction, and the epitope also exists in the virus. And so,
in the vaccine, it's weakened.
Right. And so that's where, if you're talking about sort of long-term theoretical risks, the easiest way to kind of postulate something would be exposure to the spike protein sets in motion some biological reaction. But it should be manifest with the vaccine and manifest probably in a higher probability with the virus itself.
So if that's true, then the rational line of thinking is, if I can sort of indefinitely evade the virus through my lifestyle, et cetera, then in that case, maybe it makes sense for me to avoid taking the vaccine.
Or, well, I think
and then we can get into a question of whether that's reasonably possible for people to do, or if we're going to end up living in a world where this is going to become an endemic virus that's going to be around, and eventually everybody's going to get it.
I think it's the latter. I think eventually this is going to be like flu. We all get vaccinated for flu every year. Eventually, I've had the flu. You've probably had the flu, even though I get vaccinated every year. I think eventually everyone's going to have a breakthrough infection. Everyone's going to get this. And people who don't get vaccinated are going to get it over and over again.
So to that point, let's talk about lasting immunity, because there are two main ways in which vaccines confer immunity. One is through the existence of antibodies. And then also it's through B cells. Those are the primary mechanisms, as I understand it. B cells can manufacture the antibodies. They get trained. There's a kind of memory similar to sort of muscle memory that exists for the immune system. And so what is the sort of long-term immunity that these vaccines can confer, with the focus being able to avoid having any long-term damage or symptoms from the virus, right? Even if you survive COVID, I think one of people's main fears is that they're going to get, quote, long COVID. How effective do you think these vaccines will prove to be in building that kind of long-term immunity from avoiding those types of outcomes?
Yeah, look, it's early days, right? The data is imperfect. There's studies that have come out that suggest that people who've been vaccinated who develop a breakthrough infection are far less likely to develop the persistent symptoms that we've seen in, quote, long COVID. And we still haven't clearly defined what long COVID is. I mean, it's a constellation of different things that people seem to have persistent symptoms. I think the most troubling ones, and the ones to me that are the most directly related to the virus, are the persistent neurological symptoms, because we know that the virus induces neurological symptoms. And then when you see those persist, it really gives an indication that there's some kind of neurological impairment that the virus induces that is ongoing, that persists, that creates.
That's from the virus and not the immune response?
We don't know. Right? I mean, we've seen radiological evidence of actual atrophy in certain portions of the brain responsible for taste and smell. We obviously see impairment of taste and smell. So you're starting to see biological sequelae that seem to be correlated. So the presumption is that if you're vaccinated and you get a breakthrough infection, your risk of developing those persistent symptoms is going to be a lot less. And some of the early data seems to suggest that. And so that's another argument for getting vaccinated, even if you think, look, over time, I'm still going to be a risk for a breakthrough infection. Now, on your question of immunity, there's three different components of immunity that, at a high level, I mean, I'm going to oversimplify this. There's the antibodies that circulate in the blood that everyone's measuring to see if they're, quote unquote, immune. There's the memory B cells, and then there's T cells. The antibodies that circulate in the blood protect you against infection. So if you go out right now, you have circulating antibodies in your blood, and someone coughs on you with COVID, those antibodies are going to immediately try to attack the virus that's trying to get into your blood and try to kill it. And hopefully you have some of those antibodies in your mucosa of your lungs. That's why it's important to have a mucosal immunity. And sometimes vaccines aren't the best at inducing a mucosal immunity. That's why we talk about having an inhaled vaccine, because an inhaled vaccine could actually induce the production of antibodies in your lung mucosa. That's where you want them. So that's your first line of defense. Let's say you don't have a lot of circulating antibodies. Your level's down, you were vaccinated a long time ago, or you were infected a long time ago, and your antibodies have dissipated, because oftentimes they don't last forever, and they don't circulate in blood. So long as you
produce them in the first place, because certain people have trouble producing them, right?
Exactly.
But okay, go ahead.
Immunocompromised. So now you're dependent upon your memory B cells and T cells. The memory B cells, when they see the virus, they're going to say, I remember seeing this virus, and last time I saw it, I produced these antibodies, so I'm going to start producing these antibodies again. But it takes a couple of days for those memory B cells to differentiate and actually start producing antibodies. By that point, you're infected. The virus has gotten in, it's gotten past your first line of defense, it's infected you. Now your memory B cells are churning out copious amounts of antibodies, but 2 days later, so that helps you fight the disease, it helps you fight off the infection and keeps you from getting very sick, but you're now someone who is infected with coronavirus. And the T cells are the same way. They'll kick in and they'll prevent you from getting very sick. They fight disease, but they take a couple of days to kick in. So 2 days have passed. That means the virus has gotten past your first line of defense, those circulating antibodies. It's now in your body. It's replicating. You're an infected person. You might even be somewhat contagious. And now your memory B cells and T cells are kicking in, and they're going to keep you from developing really bad symptoms. So you're still protected. The vaccine's still doing its job, keeping you from getting severe COVID, being hospitalized, dying. But you now have what we would call a breakthrough infection.
And that actually speaks to something really important, which is being able to differentiate between symptoms caused by the virus itself versus symptoms caused by our immune response. Because the more our immune system delays in recognizing the virus, the more intense the response would be, and the more the symptoms as a result of our own immune response, right? Which is one of the benefits of having B cells that recognize the virus immediately, that are already trained to do so.
That's right.
How long does that last, do you think, for this? I mean, is there any way to know how long those B cells will be able to go on recognizing the virus?
As best we can tell, look, we're only a year and a half into this. As best we can tell, there's persistent immunity against this virus, either from vaccination or from natural infection. I mean, people who've been infected also seem to have persistent immunity. We're not seeing wholesale reinfections of people who've been previously infected. Is that immunity going to last forever? Probably not, especially as the virus continues to drift. But it's probably not going to fall off a cliff either. It's probably be sort of a gradual decay, and that decay seems to be, fairly gradual. People who've been vaccinated, yes, the vaccine-induced immunity against infection is declining, but we're still seeing that people who've been vaccinated are protected against severe disease and hospitalization. And the same with people who've been infected, especially people who've been infected with Delta. I mean, people who've been infected with Delta, probably not only are they have a stronger immunity because they've been recently infected, but they're probably going to be more protected against subsequent variants of Delta that emerge because they've actually been exposed to Delta versus people who are infected with one of the earlier strains, or even people who are vaccinated against the vaccine uses the Wuhan variant, right? So that's 2 generations removed from the Delta variant. So we should recognize that, people who are infected do have immunity. It's not going to persist forever. Eventually, they're going to need to be vaccinated, but they do have immunity for a period of time. And that's why if you look at the Israelis with the Green Pass, one way to get a Green Pass to go in places is to get vaccinated. The other way is to provide evidence that you've recently been infected with the Delta variant because they recognize that confers immunity. The problem with using such a scheme here in the U.S. is nobody would agree to provide information about their recent infection. So, it's hard to set up such a scheme here in the U.S.
So there are a lot of things we're not going to get a chance to talk about, including the origins of the virus, but I think those have gotten sufficient attention in the media. There are 2 things that I want to try and get to before we close. One is antiviral drugs, because I think this is the hope, really, that we're going to be able to treat this virus. I think in the early days, what was so scary about it was the fact that there was no known treatment. But to that point, before we talk about some of those things like Merck's recent announcement about their oral medication, I want to talk about ivermectin. And I want to talk about also the larger conversation around alternative drugs that aren't, let's say, in the pipeline being developed, and what feels to a lot of people like a level of censorship around that conversation. So 1, I'm curious what your thoughts are on ivermectin, and 2, do you feel that there is an unwarranted level of censorship or bias in the media around the discussion of these drugs, whether it was the early discussions around hydroxychloroquine or now ivermectin?
Yeah, look, I don't think that there is censorship. I think that hydroxychloroquine has been the subject of hundreds of studies. None of the rigorous studies that looked at hydroxychloroquine demonstrated treatment benefit. There's been a couple of studies that have been poorly designed that show that there may be a treatment benefit. And everyone who wants to believe latches onto those studies and ignores the randomized controlled trials sponsored by the NIH and the British. I think reputable researchers who are conducting rigorous studies studied the drug, and the studies proved negative. And so you've seen groups continue to study the drug, and then there's one study that shows a signal, and those who want to believe latch onto it. And the same thing with ivermectin. I mean, there's been rigorous studies looking at it. They haven't found a treatment benefit. There's been a handful of studies that show a potential signal of benefit, and those who want to believe that the drug's providing a treatment benefit continue to point to those studies. So, everyone chooses to believe their own selective information on social media sometimes, and the question is, do we have a truth standard? Are we willing to do the definitive study and believe it, or are we going to do the definitive study, and if it doesn't give us the answer we prefer, we're going to ignore it? The British recovery study has been the best study conducted to date looking at various therapeutics. It's a study that proved that dexamethasone reduces the risk of death in hospitalized patients requiring oxygen by 50%. We've believed everything that came out of that study. That study showed hydroxychloroquine didn't work. People who want to believe hydroxychloroquine works, believe everything else that came out of the recovery trial except for the result on hydroxychloroquine. So, that's the challenge. Where do you point? I point to the recovery study and, the bigger randomized control studies. Some people choose to discount that information.
How much do you think the debate on both sides has been driven by science, and how much of it has been driven by preconceptions and politics?
Oh, that's hard to say. I mean, I don't think this debate is unique in that there's preconceptions and politics that color how people interpret information and what information they choose to put more credence in and who they choose to trust. I mean, that's true of every debate that we have. But on the scientific issues, clearly there are there's science, and then there's people's interpretation of the science. I think it's fair to challenge people's interpretation of science, even the experts. You can even challenge the experts, the so-called experts, on their interpretation of the science. But a definitive study that says that something doesn't work, that is a rigorous piece of evidence. That's not interpretation. That should be taken as fact. And multiple rigorous studies that say that something doesn't work, that should be taken as, immutable. And there's been multiple rigorous studies done showing hydroxychloroquine doesn't have a treatment effect. And that's why everyone stopped prescribing it. I mean, that's why, when the president was struck with COVID, he didn't take hydroxychloroquine. He took the monoclonal antibodies and he took steroids. He took the drugs that had definitive evidence associated with them.
Right. So, Scott, I could definitely talk to you for a few more hours. You have a hard stop. I'm going to have to end it here, but there's so much more I want to talk to you about. Like I said at the top of the show, I've really appreciated the example that you've set. Lots of people are going to be unhappy with this interview because this is such a divisive issue. We didn't have enough time to talk about all sorts of other things. But I really appreciate your approach as a professional. I've admired it, and I've looked towards it as an example. So thank you so much.
Thanks a lot. Who's going to be unhappy? I don't know your audience that well.
No. Lots of people will be unhappy. And my point is people always find my audience. If you look at the reviews of my podcast, I've gotten criticisms from people on the right and the left feeling I don't criticize Trump enough, or I criticize Joe Biden too much, or we're not talking about the vaccine. It's true. It's just how it is. You know how it is. I don't know. Maybe you don't spend enough time. Okay. All right. Well, thank you so much for coming on the podcast.
Thanks a lot. Thanks for having me. Take care.
So for all of you out there listening, I'm going to keep this mic running hot, and you can join me on the other side of this episode for my thoughts about the conversation that I just had with Dr. Gottlieb and so much more that we didn't have a chance to get into. And for those of you who are new to the program, Hidden Forces is listener supported. We don't accept advertisers or commercial sponsors. The entire show is funded from top to bottom by listeners like you. If you want access to our premium content, which includes overtime segments with guests, special one-off episodes, afterthoughts like the one that you're about to hear today, as well as the transcripts and rundowns to this episode and every other episode we've ever done, head over to hiddenforces.io and check out our episode library, or subscribe directly through our Patreon page at patreon.com/hiddenforces. There's also a link in the summary page to this episode with instructions on how to connect the overtime feed to your phone so that you can listen to these extra discussions just like you listen to the regular podcast. So come join me and thousands of others on the other side of this episode. For more information about this week's episode, or if you want easy access to related programming, visit our website at hiddenforces.io and subscribe to our free email list. If you want access to overtime segments, episode transcripts, and show rundowns full of links and detailed information related to each and every episode, check out our premium subscription available through the Hidden Forces website or through our Patreon page at patreon.com/hiddenforces. Today's episode was produced by me and edited by Stylianos Nicolaou. For more episodes, you can check out our website at hiddenforces.io. Join the conversation at Facebook, Twitter, and Instagram at Hidden Forces Pod, or send me an email at dk@hiddenforces.io. As always, thanks for listening. We'll see you next week.